All right. Well, it's certainly fun to be here. It's also a different kind of experience in terms of trying to teach like this. Does the volume seem loud to you guys? Or is it just very echoey up here? Hopefully, maybe it's just that spot. So I'm going to be doing this off of my iPad, so it'll be kind of like Lindy's talk, not nearly as beautiful and elegant, but I suppose the same could probably be said about myself versus Lindy. But I didn't realize I'd be 20 meters away from the board off to the side, but hopefully it'll work out fine. I will also, once I'm done writing on these, I can give the slides to the organizers. So if you want to sit back and take in things and don't feel the pressure of writing certain things down, I can make that available. On the other hand, it is a part of one version of learning to take things in as you go. But at least that'll be an option. So I'll certainly be doing kind of, I guess, a mixed version, both in terms of educationally, where there'll be some active stuff. So as to say, as it goes through, I'm also kind of going to be hybridizing the like research talk and teaching stuff that I'll be bringing in little threads of where somebody in my lab had studied this sort of thing as we go. And then there'll be, for the tutorial part, I'm also going to kind of blend, that won't get started today, but I'm going to kind of blend the lecture and tutorial when we get to that tomorrow, and I'll be doing we'll do some simulations in R, and so I'll make sure that the thing probably to do is make sure you've got like R and RStudio already on a computer or sit next to someone who has that set up so that we can play some games on there tomorrow. So I'll go ahead and get started. So first off, who in the world am I? I was lucky enough to be able to have Deepa work with me for a stretch of time and thus explains the invitation. That was back when I was at Harvard. Now I'm at the University of Idaho, which is over between Washington and Montana. We were just discussing how far north and also how far west it is compared to a lot of people's expectations. Though where I flew from is actually from Copenhagen. I'm on sabbatical, which makes doing something like this possible. And so even further north, but thankfully a lot closer to here. So my flights were not as taxing as they could have been. And just in terms of what I work on, you might find a word somewhere in there that interests you, hopefully more than one if you're at this meeting. I've been interested in microbial physiology, like actual molecular microbiology, how do things work? And that's kind of where I got started in terms of PhD work. And then in my postdoc, I brought my system and my questions to Rich Lenski's lab and started picking up experimental evolution. And then when I started my own group at Harvard, it was really when kind of latent interests in quantitative ways of addressing questions began to surface, initially through collaboration, then ultimately even some people within the lab kind of taking those sorts of things on. And so my favorite stuff to work on would be kind of right in the middle of all three, things that couldn't be kind of cleanly described as just one field or the other. And so I'll certainly aim to blend in various aspects of things that have gone on before into what we're doing. I think I provided to Deepa, I wrote, a friend of mine asked me to write a book chapter. I tell people don't ever bother writing book chapters because they don't count as real papers, et cetera. But at this point, I don't really care about papers. I'm tenured and whatnot, so that those sorts of things don't matter. And it was only because of my good friend I decided, all right, fine. I'll write it, what do you want it on? Evolution of methyllotrophy, like in the lab. Like, well, I'm the only person who's done it, so you want me to talk about kind of, like a personal story of kind of how things went? And the answer was yes. Okay, I like that. That I'll go ahead and sign up to do, because then it's also a good intro for people coming into the lab. And so if you want to kind of see kind of a bit longer version of some of these questions, and kind of you get to hear about stars like Deepa and the things that she's done in there, that's also available. Oh, yeah, here it is. So, and I think it certainly brings up one of the fun things I think that makes experimental evolution a little bit different than a lot of other things, in that you set up an experiment with some kind of big, grandiose question in mind, and almost invariably the interesting things that come out or pop out the side in some other way that you hadn't possibly expected. And I think that's one of the things that makes it a lot of fun. And I will probably not actually talk too much during the different sessions on the things we're currently doing, except for at the end. My research talk's on the very last day. So if there are people who are interested in talking about those sorts of things sooner, the few of the different projects we have going now, some of them are more physiological in terms of looking at things like a new regulator that senses a toxic molecule and actually interacts with the cell's translation machinery to try to protect the cell. That's been really fun. One thing that you'll hear about on Friday in the research talk, but also is true of a different project, is studying phenotypic heterogeneity, how genetically identical cells can actually behave quite differently here in the context of metabolism, and taking advantage of the fact that some metabolites, those that are stored within cells, which kind of become like their belly fat, that they can store up extra energy, you can see it. And so you can link metabolic process with individual cells or individual genotypes. And it has kind of a biotechnological spin. We have a project that's actually somewhat similar to some of the work that's going on in Deepa's lab, looking at natural variation in methylobacterium and trying to kind of at least start to ask the question, why are there so goddamn many methylobacterium types on any given leaf? Why don't you just have one or three? And the same thing in like Bacteroides in the gut. You don't have a Bacteroides, you have this whole fanned out variation, both at kind of genus level, species level, strain level, et cetera. Is there actually, ecological mechanisms that keep them all in there? Are they differentiated in terms of traits? Can we tell how the traits relate to genomes? All those sorts of questions. So that's certainly another aspect. And the things that are probably the most close to what I'll be talking about today. So I guess some of that does involve frequency-dependent fitness, which I'll get to in a later lecture in trade-offs. But looking at if we could mathematically describe fitness landscapes that selection is largely acting upon, could we then have some predictability, actually have some probability, some guess as to the likelihood that the first beneficial mutations would occur in like these targets or those targets? And if, almost like what we just heard, conditioned upon if you had this first mutation, then how does that change the probability of the next mutations versus if you had mutation B, how does that change kind of the path of things? And set up within kind of engineered metabolism and some of the stuff that we will be talking about tomorrow. So I'll go ahead and stop there for now. But like I said, if any of those things happen to ring bells, since I largely won't be talking about those through this, I'd be happy to when we have our different time. So I had changed the ordering of things so that I try to get a little bit better set up by the time I get to the tutorial section. Decided to instead get started with epistasis. And then we'll go to evolution and metabolism, and we'll look at epistasis in that context. And so I wanted to make sure to kind of get to this first, and then we'll kind of keep going from there, trade-offs. And I'm also not exactly sure how much I'm going to get through on any given time. And I think certainly please feel free to interrupt and ask questions as we go. I think that's going to make this the most fun. So epistasis. So what is this? So this is simply the idea that any given mutation can have a different effect upon a phenotype, depending upon what other mutations are there. Right? And so this is a very simple idea, but the idea that any given mutation is not just going to have some effect upon a cellular phenotype, regardless of background, that different mutations may affect what a given one allele versus another allele will cause. And this can really be on any phenotype, but a lot of the time I'm going to be talking about the epistasis upon fitness. But like I said, it really could be anything. It could be epistasis on tail length or hair color or whatever. So to illustrate this in a simple version first, imagine two mutations, X and Y, each individually improve things by 50%. So it's very, I'm going to use W for Malthusian fitness quite a lot. And I'll talk a little bit more later about that. But it's essentially the relative reproductive success of one type versus another. And so if you had this sort of scenario, which is another way of writing what I just said, if each of these two mutations makes your fitness 50% higher, then the question. Mine doesn't squeak, which is nice. What would be your null expectation, right, for the double? Right, if each of these things have a 50% effect, what do you think is going to happen there? And so you can think about that for a second, and maybe I will go ahead and do one of the things that annoys students. You can turn to your neighbor and see if you have the same thought for what you think that double should be. I'll come back in 20 seconds. So what numbers come to mind for the double? Is a possibility. 2. All right. Any other numbers? With a less than or equal to. All right. 2.25. Any other values? 3. Any other values? Is that explained? 1.5. Oh, this is great. If I just keep asking, will I get more numbers? Any more math that anyone possibly came up with? 0. Wow. You want 0? You want 1? 1. Okay, well, lots of possible choices. So in principle, of course, in real biology, it could be anything, right? It could be 0. It could be that these are individually beneficial. We'll talk about this in a second, how we would define that. And yet you put them together and you're dead. Totally possible. Like it could be 15 or something like that. They could be together phenomenally synergistic. It could be anywhere. But the question is, what kind of null expectation might we have and why, right? So I can, if you define what each of these are, this, I guess, you could define as going back to the ancestor or something like that. Or you took one selective effect of X and simply took away the selective effect of Y. This, I guess, might be the largest of the singles. Here would be, I guess, fitness of A plus fitness of B. But the fitness already has 1 built into each of them. So that's 1 plus the selective effect of X plus 1 plus the selective effect of Y. But these middle two, I think, are fairly easy to think about. This would be like 1 plus the selective effect of X plus the selective effect of Y. And here's 1 plus the selective effect of X. Its fitness times 1 plus the selective effect of Y. And I would say usually it's been these two that people have considered most frequently. Either that things are going to have some additive effect upon fitness, or that you're going to have a multiplicative effect. And I'm going to try to defend why I think one of those, at least for the sorts of things that we're talking about, makes more sense here in this situation. Right? And really this becomes a question of which null model for independent effect to use. And some might say, and I've certainly heard this many times, well, for small s, if we're talking a percent or two, then 1 plus sx times 1 plus sy, well, that's almost the same as 1 plus. Right? That they're, if we're talking about 1.01, you're going to get almost the same answer whether you add them or you multiply them, right? And so, when people very commonly are looking at genetic variation, yeah, there is an echoey spot right about there. When they're looking at genetic variation, then they'll often think about, like there's many traits that contribute to the size of a tomato. Let's just kind of imagine them as having nice additive effects. And I think the main reason for doing that is it's just mathematically simpler. And as long as you think that they're all going to be fairly small, then fine. Of course, it breaks down though a little bit, because even if they're individually small, if you have 50 such effects, 1.01, to the 50th is different than 50 times 0.01 added onto 1. But needless to say, when it's small, they're fairly close. And so, like I said, the idea then of epistasis, if we were going to calculate it, is the deviation from independence. It's usually epsilon. And so here's two thought questions that I like to use to make an argument for why I like one more than the other. So, those of you who like plants, we can think about successful seedlings as a trait. Or we can talk about, for those who like biochemistry, enzyme activity. If you don't like either, too bad. Right? So let's imagine two traits: number of seeds produced and the proportion that germinate. And imagine that they're two independent things, right? That you could have an allele that makes 50% more seeds, and you have an allele that makes your germination rate go up by 50%. And if you assume, and this is the assumption of independence, that these two things are completely independent, that they don't cross-interact with each other, now what effect would you have, right? The total germinated seeds, right, is just going to be the number of them times the proportion that germinate. So we'd have something like this. That either one of them is going to boost the other number, will boost the total number by 50%. So if you've already boosted it by 50%, you need to boost the total now to 1.5. The same is, so that's successive life stages. And it's really easy to imagine, all right, fine. For that sort of scenario, I can see how a multiplicative model makes sense. But what if there are two traits that are occurring kind of simultaneously? Well, there's something like enzyme activity, where we could think about expression level. The concentration of an enzyme. That's not its expected value or anything fancy like that. Or we could think about the activity per molecule. K-cat. Same story, right? What if you had 50% more enzyme activity? You'd have, if you increase the expression level by 50%, then the total activity would go up by 50%. If you increase activity per molecule by 50%, the same is true. But what if you did both, right? What if both things were true? Then you're going to end up multiplying these two together, right? And your total activity, which we call Vmax, and I'm also, in this way, introducing some of the points I'm going to have to make tomorrow, so that's good, would just be the enzyme activity times Kcat. And again, you have a multiplicative sort of scenario. Right? And so it's not, again, this is, it may be a somewhat subtle point, but I think it's important if we're going to begin talking about deviation from independence and kind of, kind of the boring sort of situation, what kind of expectation we should have and why. And so this is the reason why I think for these sorts of scenarios, I think these thought examples lead to a multiplicative null model. If there was independent effects. So, how can we look at this and think about this? Do a different set of numbers. I also very strongly agree with what Lindy was saying. It's nice having some tangible numbers. It certainly, I think, makes me feel good about thinking about these sorts of concepts. So if this were true, if one mutation increased fitness by 10%, the other by 20%, what then would be the double mutant, the null expectation? Hopefully not the hardest arithmetic in the world. I think I heard 1.32. Yeah. Right. So there's different ways that we can think about this. I'm going to represent it in a few different ways through the talk, so I'm going to throw multiple of these down at the same time. So we could think about one way of writing these different backgrounds is to think of them the way that I have so far, the ancestral, a single mutant, and a double mutant. We can represent this like this, where 0 equals ancestral and 1 equals evolved in this case, in terms of the way that I'm thinking about things. It could be something else. You could, of course, have more than just two states, but for now, we're going to just have zeros and ones in our thoughts here. One way that also is useful for then seeing is the idea that then if you have them acting independent, X provides a 10% benefit. Again, regardless of background, Y presents a 10% benefit, or 20%, sorry, regardless of which one it happens on. Yeah? All right. One last way of drawing this, and then we can talk about the different kinds of things. I've heard this referred to as a lollipop diagram. This represents fitness, though to keep things simple, I'm actually going to have it be log of fitness, or plotted on a log scale. I'm going to still write the linear values. Zero, zero. All right, then colors, I may as well use them. So one thing that's nice, I think, about thinking about this graphically is if we actually have 1.32 as our null, if the actual data match the null, then it means in this fitness landscape is just a simple plane, right? It has no slope to it. We can then think about any possible deviation from that as having some bend in the landscape. And this would be true, this flatness would be true as you extend it to a third, a fourth, site, locus that you're thinking of, it's just, I can only attempt to draw this with, 2 dimensions and in implying a third. I'm not very good at 3-dimensional and implying a fourth and things like that. So we'll leave it as this. So what if, in terms of the actual values that could come out of this, you could see something like 1.5. So it would cause a bend, kind of an upward bend in this whole landscape. You could have something like 1.22 or something like that, right? And so one thing that's kind of nasty about thinking of epistasis is it's a field in which there are probably as many different names for types of epistasis as people who study it. And many of them are partially redundant or completely redundant, or have one meaning for beneficial alleles that's different for deleterious alleles, or not describable if you have a mixture of beneficials and deleteriouses together, et cetera. So it gets a little bit nasty. So I want to throw a few of these down because they're the kinds of things that certainly here, and it's worth thinking about. Anything that's more than what you were expecting, we call positive. I did not write out. I should have. Yeah. I didn't write out then. Epistasis then is, if this is it was the deviation between, the data and what you see for the null hypothesis, right? And so it's like what you actually saw for the double minus what you thought you were going to get for the double, given the 2 singles, is going to be what you get for epistasis. So if that double, like 1.5, is larger than the 1.32, it's positive, obviously, the epistasis value. Shockingly, then any value less than 1.32 is called negative epistasis. There's another way of, yes, please. Mm-hmm. Mm-hmm. Yeah, so there are definitely aspects that can lead to some complexity here quite easily. One, probably maybe one of the simplest is if here we're taking kind of a ground state, like an ancestral cell, and we're kind of adding alleles to it, et cetera. And we have a concept of the single effect of alleles are this upon that state. If you're taking a population that has a lot of different variation, there isn't any like kind of single kind of ancestral state, and you have the ability to test, well, on this type that we're going to leave everything else the same, what effects do they have? Then you need to turn to things like, well, the average effect of this allele across many backgrounds is something. And then like that is different, right? It's not, here the only like variation you have, when I say 1.1, like really there's a measurement, right? And 1.1 might be the mean, but I've got some, some uncertainty around that, some measurement noise. Whereas if you're taking an average effect of allele across a population, there's measurement noise, but there is like actual epistasis, et cetera, going on. And so it does become a little more nuanced and so maybe let me continue through and see if you still have questions about it. But I do think it's a good point. And I think it somewhat reflects what kind of experiments you're able to do and what kind of questions you want to ask in terms of kind of how exactly some of these things can be defined. Because certainly, like in a human population, you don't have this possibility to do the experiment where we're going to take my genotype and then I'm going to just change one allele and find out what happens, then just change another, and then like it does, you can't do that, and thus, yeah, the way of thinking about it is a little bit different. Yeah. So there's another interesting boundary. If you take the highest of the two singles, so as long as that double mutant is at least 1.2, above that, then mutation X would be beneficial on both backgrounds tested. Mutation Y would be beneficial on both backgrounds tested. Just the magnitude of how much effect they had alone versus in the presence of the other mutation went up or down. So anything like from that's above 1.2 to up to the ceiling is just going to be affecting the magnitude of the epistasis, but not the sign. And so it'll create kind of an upward curvature, but there's no like downwardness to it. And so this, because it keeps the same sign, this is also sometimes referred to as magnitude epistasis, which is often kind of the boring little brother of the one that's coming up next in some people's minds. Though I would say that's not quite fair. But you can also have epistasis where it changes the sign. Let's go back to and this can have various sorts of shapes because it's a lot easier to draw them this way. Let's just do this. And I can do this, which you can't. All right, so this could take a variety of forms. You could have like what we started with, but now maybe we could have 0.8 or something like that, where it's below the single. In fact, it's below all of the other ones there. That would be something that would show sign epistasis. We could have this sort of scenario. Each one is actually individually deleterious, but together it's fine. You're kind of back to where you were. You could have this sort of scenario. Where from the perspective of mutation Y, it's beneficial on both backgrounds, but from the mutation X is beneficial on one and deleterious on the other, right? So it could have many different scenarios going on. This one has sign epistasis for, which one flips? X. These two see sign epistasis for both. And there's some interesting kind of particular cases of some of these. It's like this one, where you have two mutations and combined, they're kind of like particularly bad together. There's an extreme case of this. Imagine all combos except the 1-1 are viable, but but 1-1 is lethal. Right? So each individual mutation is tolerated, may even be neutral, may be beneficial, could be whatever, but then you put them together and you're dead. There's a name for that. Anybody happen to know what that's called? Some biologists love this. It's called a synthetic lethal. And it often implies something, like imagine you have two different biochemical pathways, maybe the simplest might be two isoenzymes for a given step. You have just this copy and that copy. And you could delete one of them, and we'll talk about tomorrow even why deleting one of them might have very little effect. You could think about this as being the same locus and dominance and recessivity, which we'll talk about. You could delete one copy of the gene on one chromosome and be fine, or the other, but the both in your SOL. And so this is certainly one type of sign epistasis that's fairly common. This one is actually also quite common. To have what are sometimes called compensatory mutations. In this case, they're reciprocally compensatory. Right? It seems at first a little bit weird. Well, like one mutation was kind of awful. The other one was pretty awful, but combined, you're just fine? Like, what's a biological example where that could actually be true? Can you think of something where that might be true? You make one mutation, pretty bad. The other one's pretty bad. Maybe even lethal in some cases, but combined, just fine. Mm-hmm. Sure. Two molecules or even within a molecule, right? So you can imagine a point where within a protein, or this is actually very common in RNA structures where you have stem loops. And so if you imagine something like this, where there's some nice, well-behaved little stem loop, right? So you can imagine mutation X and mutation Y that, let's say, goes to this sort of scenario, TGA, TAA, or to TTA, TCA. Either of those have broken up that middle pairing, but if you now simply do the other mutation, you get to a new pairing, which might, for all intents and purposes, be just about the same, right? And so, fine, this could happen. Does it have any relevance? So one thing about this is this, I think, provides an example where you're fine now, you could be fine 2 mutations later, but in this case, like any step that's kind of in between is going to be problematic. This is often thought of as crossing a fitness valley. because you have to go through these low fitness intermediates. And it turns out this is a major reason for why changes are so slow in things like 16S RNA. And so people are probably, hopefully you've heard of ideas like, I'm not sure how much phylogeny, et cetera, went on last week, and ideas like molecular clock, that you have some changes that occur reasonably slowly, and some that happen more quickly. One of the nice things about 16S is that it's a chronometer that actually has kind of hour, minutes, and seconds hands on it, because you have parts of it that are not part of these stem loops, and they change relatively more quickly. But these have a much slower time scale at which they change. And one of the major reasons is it's only when this relatively rare event of being able to cross this fitness valley can occur that you manage to get from a GC to a TA. And so it creates a much greater kind of sieve for such things, and so they occur substantially less frequently in terms of getting fixed along lineages. And so and the same is true with proteins. In fact, that where you have, whether they're between partners or like I said, within a protein, places where you have shared residues, not only do they kind of co-evolve with each other, so you see coordinated changes on a phylogeny, it causes both of those sites generally to evolve more slowly, unless you've got some sort of fun selection to like run away from a pathogen, and then interacting residues can actually change a lot faster than you'd expect. But needless to say, and all that ultimately comes down to epistasis. How do the mutations kind of affect each other? All right, so what does this actually look like? Ironically, it's only been relatively recently that people have begun being able to do these sorts of experiments to test these things. And so how, at least from the point of view of adaptation, and a lot of what I'm talking about here is kind of in that light, we can look at things like evolved alleles of proteins and ask how those mutations affect each other. You can ask questions, like, how the mutations. And you can ultimately ask, is it strong enough? Is the way in which mutations interact with each other enough that it might actually change what it takes to be able to get to different positions? So the example I'm going to bring up here is one from Dan Weinrich. And it's kind of become the classic textbook example for this sort of thing. And so he looked at an enzyme, beta-lactamase. Anybody know what that enzyme does? Anyone who's not shy know what that enzyme does? So it's, anytime you see ACE, it's something that does cleaving, and it cleaves beta-lactams. This is what provides penicillin resistance. And so it's an enzyme, and it cleaves beta-lactam. And thus, like many proteins, there's kind of at least two things that have to happen. It needs to be able to fold to be able to make a protein, which is true for the majority of proteins. There are some that are unstructured, but we won't go there today. And then it has to do what it needs to do. And in this case, it needs to be able to bind and catalytically cleave its product. And so compared to a wild type allele, after a period of selection, they got a mutation, an allele that was much, much more resistance. And it took a lot of mutations, which seems a little bit funny. I just told you this enzyme cleaves beta-lactamases. Why would you need mutations if it already does this? As many of you probably know, that often resistance arises quickly, and then with things like beta-lactamases, people have played the game for a long time now of trying to make a molecule that still has the antibiotic function of penicillin, but is not cleaved by beta-lactamases, which then, of course, just creates selective pressure for cefotoxin resistance, and then you can try to make yet another version that still inhibits cell wall biosynthesis, yet can't be cleaved by all of the classes that are out there, et cetera, and you kind of have this war sort of situation that escalates. And so it was a perfectly good beta-lactamase against other beta-lactams, but against this new version, cefotaxime, they were trying to find out what sort of mutations could occur. It took 5 of them to get an increased resistance, and it was a pretty big increase in resistance. There was a 40,000-fold increase. They were good in terms of making cefotaxime. It really was not well bound by the original. The sad thing, though, is 5 mutations later, which is not that many necessarily, especially in light of selection, and it's very good at being able to chomp it down. So what would you do if you wanted to look at epistasis and actually measure these sorts of things? Do we even need to worry about it? Is it so weak? We could just mainly think about the single effects or the average effects or things like that. And so they took a very simple strategy. They constructed all combinations. There's 2 alleles, 2 states at each of the different ones. So there's 2 to the 5th alleles. And these happen to have been 4 non-synonymous. So things that actually changed the amino acid sequence. And 1 was a promoter mutation. So it increased its expression, which is also a very common sort of thing. And then, of course, you want to assay resistance. And for this, they measured the minimum inhibitory concentration. MIC. It is both spelled MIC or sometimes pronounced MIC. I usually use MIC, I guess. And what this looks like generally, and so this is definitely it's a little bit of a problematic definition. Certainly, if something has a higher MIC or a lower MIC, that probably does tell you something about what's going to occur in real life. But in a patient, there's going to be a whole range of concentrations in different parts of the body. It's going to change temporally with time, et cetera. These MICs themselves, they definitely tend to be a more of kind of an operational definition because you need to define kind of, in a particular condition, and because people are lazy, this is usually something like 37 degrees, LB broth, 96 well plate, whatever's the easiest thing to do. It's not usually in a realistic setting, and 24 hours later. And it needs to be below some threshold of growth. And unfortunately, if you change any of those things, if you wait a little bit longer, if you set your threshold a little bit differently, you have different conditions, you get different kind of answers. But effectively, you get something that looks like this, where you can run some plate and have different antibiotic concentrations and then different strains. And in this full combination. Good enough that you'll get, for any given strain, you'll get growth up to some point. I'm kind of just filling in which wells would have turned cloudy by 24 hours. So then it means your MIC values would be the lowest concentration where you got no growth by 24 hours. Make sense? So this is really dumb, simple work to do, even though, like I said, it can get a little bit touchy. So, great, you do this, what do you get? So you get this is the picture from Dan Weiner's science paper, and the first thing that might seem confusing, I said they made 32 alleles, and this figure does not show 32 alleles. And if also, we're going from the ancestral allele over here to the highest evolved allele. And I told you that there's 5 possible mutations. So as you guys can probably figure out, well, from the first step, shouldn't I have 5 possible steps? And then after that, 4 possible from each of them, and then 3 possible, and then 2 possible. And because it's a limited set of choices, you'll if you've made all those choices, you'll always end up at the top. And yet, it doesn't look like that, right? Why are they hiding data from us? There's a reason. There were 3 other possible single mutations. But these 3 were all had equal or lower MIC than the ancestor. And so what they've done is they've filtered this whole chart. This shows only steps that would be driven by selection. So as Lindy is going to tell us very elegantly, if it was a neutral allele, how lucky you'd have to be to take one of those steps. And if you were a deleterious allele, how damn lucky you'd have to be, at least in this environment, to rise up. Now, in smaller populations, maybe the neutral ones are going to have a little bit better chance. Things like that. Also, it's going to depend upon things like environmental change or mutation rate. I mean, certainly other things are going to influence this. But the main concept about this thing is if we look at what's going on here, we can see actually a couple things that can be said. So this mini combo is missing, means that there's some flat or downward steps. Okay. Yeah, so like to give an example of where there's not a downward step, this triple, that one that says 32, by the time you get to that step, every one of its next possible mutations are all beneficial and are all shown. So if it was unlimited, like I said, you'd have 5 and then 4 and then 3, et cetera, from each node. But what else? So if you kind of take in and admire the grandeur of this kind of data, what else though is true about this landscape? There's kind of one other qualitative point I think that's fairly important about what was seen in this case. You think about it from the idea of fitness landscape. Evolutionary biologists are optimistic. Our landscapes point upwards. Physicists think about energy landscapes, falling downwards, right? If you were to release, if we think of the downward step, for example, if we release marbles onto this landscape, where are they going to end up? Huh? Sorry, I couldn't. Well, so these selective differences are kind of like the potential. They're saying kind of how strong selection would be driving towards different things. So there's, I don't know, about 16 or something alleles shown up there. Which one does this imply? And if you play, which is also a slightly ridiculous game, that you can only make these 5 mutations, nothing else is possible in the protein or the rest of the genome. which is probably the biggest concern about this sort of thinking. But imagine these were the only steps you could take. Where would the marbles end up? Are there multiple energy wells? It would be the global, right? So energy well, fitness peak, same idea, right? In this case, it's a one peak landscape. There is only one best possible scenario, and there's no intermediates that are kind of locally trapped, right? Imagine that you get to this 362, and if you did that, any mutation coming out of it was deleterious, that there was sign epistasis, that you got there during positive steps, but you can't move out. Then there's going to at least be some probability that you took those roads, and that's where things would go. All right? And so at least for this landscape, it did have a single peak. All right. But the fact that there's many combinations missing, even though there's a single peak, it means there's relatively few paths. Because there otherwise would have been 5 choose to 120 potential paths. I think it was something like 18 were possible. And some of these are given thicker arrows. You can make some various population genetic assumptions about what's going to go on here, how many alleles would be existing in a population at the same time, et cetera, and begin to try to assign some probabilities of likelihood, things that are much more down the road, where I'm guessing Lindy's going to take us, to even say, all right, you could have gone like all the way through this step and up, but the chance that you actually did that, because you would have gone through like this 32 instead of that 362, like that 362 would have been much more likely. So some of these paths, because of their selective advantages, would have been more likely than others, even if you only think of it from the standpoint of escaping drift and don't get as complicated as we'll probably go later in terms of thinking about clonal interference. And so there's few paths, and even of those, some of them are much more likely than others. All right, but like I said, this only shows the paths that could have been successful. I think there's another way of looking at this, and it's going to compare more to the way we'll see different type of data. And so I replotted this to show all of their data. And I looked at one allele on all backgrounds tested. So what do I mean by that? So this is showing the fold change in MIC. So of having, this happens to be the promoter allele that's upstream of the gene and allows it to be transcribed, and asking on the original allele if you have or have not the promoter allele on this allele, on that one. So if there's 32 total, and you basically have 16 binary comparisons you can make. For this one, you can take those same 32, look at a different allele, and make another 16 comparisons of, well, what about plus or minus this allele? So you can kind of slice through this data and get some various sorts of things. But you can see, for example, does this promoter mutation have the same effect on all backgrounds? Right? If there was no epistasis, if it just the null prediction, it would be a flat line. Every single dot within measurement noise would be something, say, a 0.6-fold increase in MIC. It would just be the same on everything. What do you think? Is that what this looks like? I would say this looks very strongly not like that, right? That if we were to take the average effect it's probably going to be somewhere down here. Right, is fairly modest. And we instead see several different classes of things. We've got a number here of times when it was deleterious. The cell was actually better off not expressing more of the protein. Perhaps those protein alleles were particularly toxic to the cell or something like that. And so if you actually made more of them, the net cost was worse than the net benefit. We'll talk about some things like that later. Some of these, a number of them down below too, but these especially, are definitely beneficial. There were cases where it was neutral, et cetera. So this shows what kind of is implied by the other, that there was most certainly sign epistasis. But I think another point kind of quantitatively is that the epistasis values are often much bigger than even the average selective coefficient, right? If the average fold change was like 0.4, but occasionally you're 15-fold better, right? So that the particularities of when you throw it down have an overwhelming effect. It's not just a slight modulation upon what's going on. And like I said, it can even flip sign. But there's one third point, which is why I plotted it against the x-axis. What's the x-axis? The x-axis, and I had to make it a log scale because it's such a damn huge change in MIC total. I wanted to be able to see these points. There's no fancy mathematical reason. Does it scale generically with backgrounds? So for example, if I already had a really high resistance allele, maybe their high expression would be good, but on low resistance alleles it would tend to be bad, and there'd actually be a trend in this scatter plot, right? Like it doesn't, like it seems just absolutely idiosyncratic. There's no generic relationship with how good the allele it was that it showed up on in terms of the fitness effect. And so this is one particular protein. By now, there's been a fair number of proteins where people have done this in a very kind of directed way. We're beginning to move towards being able to generate and just order from a company a large number of different allelic variants, and then use sequencing to test. You can just test all of them together in a batch and use sequencing to look at their representation through time. And so this kind of data, thankfully, is growing and growing. But I'll say, even though this was the first study, this was in 2006 in Science, those statements, strong sign epistasis, the epistasis often being much larger than the average effect, and it being very idiosyncratic with regard to kind of its background effectiveness, have held true for mutations within a given protein. So these have really held true now over at least a dozen examples. So I suppose you could say there's a very common trend, and the common trend is that there's no common trend of, it's kind of one of these f of x sort of statements, but right, that it's almost always true that there's no trend within the data with regard to these things, that there tends to be this kind of seemingly idiosyncratic, strong epistatic interactions. Now obviously this is governed by something, right? This is governed by the biophysics of proteins and what it means to fold, and the specificities of like folding pathways and interactions and the ability to catalyze and things like that. Like, it's not magic. There is a rational cause for these things, but they're so far have been relatively difficult to disentangle, and certainly don't scale with something nice like how effective the allele was before. So what about epistasis between genes? All right, so what about just simply different genes within a system interacting? These same trends hold. Right? And like I was saying, within proteins, molecules, biophysics, folding, catalysis, et cetera. Between proteins, I would say there's two possibilities, though most of the times it's the second one. Occasionally that's also, I would put under the category of biophysics of proteins. And that was the one that was brought up before about, like, say, what about two proteins that interact with each other? If it's a binding interface between two particular proteins, those epistatic interactions, they're basically governed by the same kinds of principles as within protein. They also tend to be highly idiosyncratic, very strong, et cetera. So this is, like at protein interfaces. But most of the time, they interact, so say in quotes, through physiology. That they don't necessarily touch, not that kind of interaction, but one affects how you grow, and then, say to make some, intermediate, another one affects something else. And the question is, if you, modulate both of those things, what's kind of the net effect? Or one changes a regulatory pathway in this way, one changes a regulatory pathway in that way. How do they interact with each other? That they're not necessarily, direct interactions. I should also put here like protein, DNA interactions, et cetera. Doesn't have to be protein-protein. So where a promoter and a regulator bind would also be kind of driven by biophysical interactions and also tends to be pretty idiosyncratic. So you could imagine an analogous experiment, the one above, for epistasis between genes. Or you can start with some organism, end up with an evolved strain, sequence to find mutations. Now you're sequencing a genome rather than just sequencing an allele. But these days, that's not a challenge. It used to be. And then from that, make the combos. Test fitness. And then publish in Science, like the first paper. And luckily, even though I'm being facetious, that actually did end up happening in that sort of order. Now like I said, I'm gonna be kind of blending research talk and this, et cetera. I'm gonna bring in a couple model systems. I'm gonna try to keep coming back to the same ones because I think it would be exhausting to hear the particularities of lots of model systems. That's one. And also, one of the model systems is the one that like myself and Deepa, et cetera, have worked on. So obviously we have quite a bit of familiarity with it. And the other is the most famous system within experimental evolution, the Linsky E. coli lines. So I'm going to get to those and introduce, did Linsky lines come up in the Schlatter? Great. So I can make that nice and tidy when I get to that. But I'm assuming probably methylobacterium did not come up. So I'm going to just have a couple slides just so you know what this organism is. I'm assuming you did not talk, or at least about this. Okay, great. So the particular bug, so this is like an aside from the flow, that I'm going to be talking about a few times. It's called methylobacterium. It lives on plants, as we'll talk about. There's a nice little leaf print on an agar with methanol in it. So every time you have a salad, you're also consuming things like this. It's a fairly dominant member of the plant microbiome. It can be between 3 to 30, 40% of the microbes that you find on there. It can eat single carbon compounds like methanol, but it can grow on a limited number of other things, which means then you can do genetics on these C1 pathways. And because of that, it's been the model system for the last 60 years. Almost every arrow that we know about this becomes this in terms of the metabolism going on was discovered in this system first. So it's kind of the E. coli of this type of lifestyle, things that eat single carbon compounds. And what does that have to do with plants? It turns out plants actually release methanol, that about 3 to 10% of net primary productivity is released as methanol through the stomata. And it happens once a day in the morning, generally. And it's because plant cell wall, like pectins, et cetera, are methoxylated. and those bonds need to be broken and remade each time the plants generate new cell wall or remodeling cell wall that exists. And so it releases a fair amount, and people like Julia Vorhold at ETH have shown that strains that don't have the ability to eat the C1 compounds, they can colonize a plant if they're the only thing there, but they're less competitive than an identical strain that has the ability to eat methanol. So it does actually matter. It's not just a correlation. You know, it is a part of what matters for these guys. And there happens to be a biotechnological angle to this too, that the pathways that they have happen to have very high flux through some interesting reduced compounds that could be used to make all sorts of stuff that could be converted into money. And you can use things, C1, either like biogas or geologically available methane can go this direction, and it can be cultured quite effectively to optimal densities over 150 for those who know what that means. I mean, you could grow it almost to look like Greek yogurt. Like, it's really, really thick. And there are companies out there that do this. The plant growth promotion, there's a company called New Leaf Symbiotics that commercializes the fact that it can aid like plant seed germination and plant growth. And there's also, in terms of the biotechnological, in an industrial setting, there's a company, NipBio, which I co-founded. Here's my statement in terms of that. And we're actually using this bug as a feed ingredient for, right now, targeting aquaculture. So both as a protein replacement for fish meal, since we overfished the little fish to try to feed the big fish that we want to eat. But we also think there are a number of value-added compounds that we can make in a much more cheaper and sustainable way with microbes rather than the way that they're made today. So if there's any curiosities about that sort of stuff, I'd be happy to talk about that later, but otherwise, I'm not going to be going down those roads. The interesting thing about growth on methanol, and I'm not going to belabor it for this audience, is methanol first gets turned into formaldehyde. So rather like ethanol, which gets turned into acetaldehyde before further utilization, these guys do the same sort of thing. It goes methanol to formaldehyde, and then I'll just call it a series of steps to formate in CO2, or the formate gets assimilated into biomass, kind of their version of a Calvin cycle, taking single carbon groups and adding them to a larger carbon chain. It happens to be kind of a modified amino acid biosynthetic pathway, which for them is their assimilation. And so, in this case, I mean, it's as if, if you tried to subsist on neat alcohol as your only carbon source, it would mean all the carbon that you both assimilated as well as burned off would have gone through acetaldehyde as an intermediate. I don't suggest this. I don't think that that's a good plan. And I certainly don't suggest you try it with methanol. Right? This is not the alcohol you wanted to be growing on as a sole carbon source, but these guys do it, and they do it remarkably fast. It flows at about 2 millimolar a second when they're growing at about a 3-hour doubling time. And so one way of thinking of that flow rate, very simply, is if it wasn't at steady state. Imagine you just shut off the valve for formaldehyde use, but keep making it. It would build up in the cell by 2 millimolar a second. And in a very short time, probably about the amount of time it takes for you to make this sentence, then it will cross-link all of the biomolecules in the cell. Formaldehyde's very reactive. This is why, when you see things pickled in a museum, formaldehyde is often the kind of agent that does that and can preserve the biomolecules because they can't be degraded anymore. They are all cross-linked to anything and everything else. And in the lab, we have to boil cross-linked proteins where you do formaldehyde cross-linking to later ask who was touching who. Cells have a hard time boiling themselves. And so to not pickle themselves, to not do this, they need to worry about this toxic intermediate all the time. And it ends up being important for some of the stuff that we're going to talk about. So, and where are we on time? Yeah. So back, actually when I was in grad school, this is beginning to be a while ago, 22 years ago, I found that you could replace the native formaldehyde oxidation pathway foreign pathway. And they would grow, but much slower. So it's cool. I was able to figure out kind of what role some of the other pathways were doing because I could take something that I knew was a formaldehyde oxidation pathway, and this uniquely was able to complement one set of genes and not another set of genes, et cetera. But then I realized this grow, but much more slowly, is actually a really convenient aspect for experimental evolution. And I'd begun to, I had found some of the papers by Rich Lenski, and this is many tens of thousands of generations earlier in his experiment, but they were already really cool papers at this time. And I really was fascinated by the idea of doing evolution in the lab. I still have control over what I'm doing, which I really liked, yet it's also open-ended in this really fun way. But with the Lenski lines, as we'll talk about somewhat later, like E. coli growing on glucose, ironically, this is probably the best studied organism on its kind of favorite food source. But if you were to ask somebody, what does it mean to be a better E. coli? Like what targets would mutate to grow faster on glucose? I don't think anybody would have guessed the things that have come up because they're just kind of all over physiology. It's not obvious, well, which of many possible things would you need to change? Here, this cell was going just fine before, and I totally messed it up. Right? I ripped out its engine and stuck in a different kind of thing. Right? It's like taking a diesel engine out and putting in a little tiny electric engine or something. You could make the car perhaps start to move, but you're going to need to evolve a lot of things. Maybe the engine needs to get bigger. You need to change your wiring, bigger batteries, get rid of your gas tank, da, da, da. You know, a lot of things need to happen. And even though it's kind of complicated enough to be interesting, it focuses, hopefully, selection most strongly upon a particular part of physiology and the stuff that I wanted to study. And so I realized that this might be a good starting place. And so I took this bug and myself and wife to Lansing, Michigan. And so as a postdoc, started experimental evolution with the engineered strain. And so here's kind of a time course of growth rate. If we extended this up even a little bit further, like the wild type would be kind of up about there. It's about 3 times faster. And so replacing the pathway dropped to about 1/3 as fast as it could grow. But you can see that over, in this case, a time scale of 900 generations, they've improved quite a lot. And things that are very typical and perhaps were already mentioned, very typically the improvement was very fast early on, slows down later, but is still continued. Unlike the Lenski lines, this has actually a substantial amount of across-population variation in how much they improved. That some of the lines are almost twice as improved as others. Is something that they didn't happen to see as much in that experiment. Anyways, lots of kind of interesting things to say from that perspective. Just to give a population genetic feel for such things, each transfer, this was not done continuously. So this was batch transfer. With 1^64-fold dilution. And so 1^64-fold dilution means you're doing 6 generations per transfer. And population size, for example, the final population size on the order of about 1 or 2 times 10 to the 9th. Right, so a fair number of cells. This is only 10 mils of liquid, but you have pretty good sized populations. Not infinite, but certainly big. Pretty good size. Thus, for different processes, effective populations are about an order of magnitude less than that. Let's see. And this, when I say relative growth rate, what I'm talking about, just to make sure that that's clear, if we put the log of the number of cells during exponential growth, that's going to be linear until they run out of food. They stop actually rather quickly. And so when we're plotting growth rate, we're taking that slope from the R in the growth rate equation is what I'm talking about with relative growth rate. And so actually, since Christian Slaughter was here, he talked about how to, like, fitness competitions. Yes, largely. So rather than kind of the broader scope of things, we had, when we do, yeah, please. Yeah.
Just wondering, with short times, all these trajectories look sort of similar, and they are more divergent at long times.
Kind of. I would say, I mean, even on the, I would say even relatively early on, there was a fair amount of spread. More actually, even on the early timescales, more than like the Linsky lines had. One thing is like the relative scale was much larger. They improved, like the Linsky lines, their fitness got up to about like maybe 1.8 or 2 after 60,000 generations, whereas ours got to that kind of thing more in the like, but like it depends. You know, the TIM1 beta-lactamase improved 40,000-fold with 5 mutations. You know, it just depends upon kind of what's, you know, a reasonable range that's possible. So I don't know if I would say too strongly that they stayed close and then spread. We know something about some of the mutations that were going on here. There were classes of mutations that were so common, they were probably driving the average fitness gain in all populations, but then in many cases they lost out and other things swept by them and were more successful. So then it might drive kind of relatively more similar dynamics early on, and then, you know, like whether they did or didn't pass muster later led to more of a spread, because those didn't tend to be the winning lineages, perhaps. Yeah. Yes?
So, when you look at the number of generations, basically means that initial population size was larger, so the selection force was very high. So if you had diluted it with more fold of dilution, that's like number of generations
Yeah.
higher
Mm-hmm.
per transfer, this would be the same trend.
So that is a good and somewhat tricky question. So if instead of doing a 2 to the 6, we did a 2 to the 12th dilution or something like that, how would it be different? Let's imagine for now, at least, that wouldn't have affected their physiology at all. There's one dirty detail that people don't tend to think about when they do these kind of, because people have done exactly this sort of smaller population size, larger effective population size kind of thing. It is that sometimes that actually affects their growth. So the selective coefficient of the identical mutation could be one thing in one environment, one thing in another. So let's imagine that wasn't true. And all selective coefficients would be the same under either treatment. The major effect, you know, this is, you're still, unless you went really tiny, you're not going to be into the regime where neutral or deleterious alleles would be affected by drift. I mean, you'd have to get to very small bottlenecks for that to be likely. But you will tilt the spectrum of mutations that would be successful. Are you going down clonal interference road later, I assume? Yeah, so let me largely wait and pass the baton there. But if you went to you did the opposite, let's say you did massive VATs, and so you have a huge population size, then you have the potential to draw from the best possible mutation that's out there, no matter how rare it is, or double mutant or triple mutant, et cetera. It's just going to be, it'd be a greedy search amongst kind of this huge kind of possibility, of things, only tempered by the fact that it would take time for it to kind of sweep through. As you get to smaller and smaller populations, generally to be a winner, you need to be amongst the biggest mutations that are likely enough to occur given your population size. And as your population size gets tiny enough, that likely to occur part begins to matter more and more compared to, are you necessarily the biggest and best possible thing? And so it definitely would tilt things. We have done some experiments in that sort of vein, different system. And we definitely saw these sorts of events occur. Yeah, no, it does have, you know, it can have a pretty profound effect. Yeah. And because of things like epistasis, it can actually, if you know, it won't just necessarily slow things down. If by taking some mutations you change your whole future path, you know, it could even, you know, greatly affect whether you get a wider variety of outcomes or a smaller variety of outcomes. Yeah, yeah, certainly. Great question. And so the timer has gone off. Let me see. If I can just finish the next slide, and then I think would probably be a fine time to break, and I'll pick up from there. That sounds fine. So this chart that's up there was showing growth rate. We also can assay fitness in competitions where it's a pairwise looking at change in frequency. There's many different ways to do this. We use strains with fluorescent proteins, because then instead of us doing the counting, we use a flow cytometer. You can get 50,000 cells in a minute. And that's nice for a couple reasons. It only takes a minute instead of you sitting there counting, counting, counting. But frankly, the thing that matters the most is you get good numbers, because you're sampling you know, two things. And if you sample 50,000, there's not so much, you know, statistical noise in the counts that you get. When you sample on a plate and you've got 100 colonies or 50 colonies total, there's just a lot of sampling noise. And so unfortunately, like a lot of the Linsky literature, because it's all been counting a small number of colonies on plates, the sample noise, you know, the variation from that is often, you know, on the same size as the thing that you're trying to study. And so it really becomes a problem. This we can assay very nicely and get, like here's a neutral marker, you know, being compared to itself. We can get, you know, pretty tight estimates of fitness by measuring a large number of cells this way. And then when we go to calculate fitness, when I've been saying like a fitness of 1 or 1.2 or et cetera, we're looking at kind of what was the fold increase of, and so these are the ratio from the flow cytometry data of one strain versus the other, kind of spread out over the entire growth. And so it's kind of funny. It's taking, like if in reality, if you had two lineages growing, and let's say one grew fairly quickly, then food ran out, and the other one grew slowly, but they both stop at the same time because they're in the same flask, but food runs out. By taking these ratios, like you're actually making the measurements T1 and T0 out here, and this is basically ignoring all the details in the middle, and it says, well, effectively this has done that, and this has done that, and we're taking a ratio of those slopes. And you're kind of, it's the average selective advantage per generation. Right? And so in this case, because it's a 6th generation experiment, that's where this number 64 comes into play. And so if you're 10% better, then that gets amortized, it gets spread out over the entire thing. All right. So we will get to epistasis across the genome then on the next time. I guess it's lunch, yeah?